The discovery of DNA structure is one of the most important discoveries in biology. Watson and Crick famously understated this in 1953 when they said, "This structure has novel features which are of considerable biological interest." Of course, we now know that DNA is the carrier of our genetic information, and without DNA, our life would not be possible, at least as we know it. To understand the structure of DNA, I want to actually walk you through the discovery process for our DNA structure. The discovery of DNA structure began in the 1800s. Friedrich Miescher was the first one to isolate DNA, although he referred to it as nuclein as he extracted it from the cell nucleus. Now, of course, at the time, he had no idea that this was what carried the genetic information. It was actually thought that proteins might be the carriers of the hereditary information. Through a chemical reaction-based approach, other scientists then began to propose structures for DNA. So through hydrolysis reactions, they were able to identify that the nuclein contained phosphate, pentose, and a nitrogenous base. Now, of course, this is what we focused on when we looked at the individual monomers of our nucleic acids. Through further chemical analysis, it was then determined that the phosphate was linked together with phosphodiester linkages. So again, using this esterification through alcohols on our sugar groups attached to that phosphate to form our phosphate esters. They were also able to identify the exact pentose. So they found our ribofuranoside. Again, of course, remember that furan is just referring to that five-membered ring shape. And of course, we have our ribose or deoxyribose as our primary sugars here. Our modern picture of polynucleotide structure is actually quite similar to what was proposed in the past. So again, we have our phosphodiester linkages between our sugars, linking our nucleotides into this polymeric structure. Of course, we can be a little bit more specific now. We know that it's occurring between our 5' and 3' alcohols on our sugar rings. And of course, this results in our negatively charged backbone of our DNA due to the negative charge of our phosphate. So each of our phosphates linking our sugars together will be negatively charged. Now, this backbone is relatively stable, which of course is very important for our DNA since it has to exist over a long period of time, safely packed inside our nucleus, where it can then be copied to create our various proteins. Now, as noted in that experiment, hydrolysis can be used to break apart our DNA structure. So esters, of course, can be hydrolyzed in the presence of base or acid. But for our DNA, it's relatively resistant to hydrolysis unless you have enzymes that are present that will be able to catalyze and speed up that reaction. Because our phosphodiester always forms between the 5' and 3' alcohol groups, our DNA always forms linear polymers. So there are no branches or cross-linking. It's just strictly a linear polymer following this 5' to 3' direction. Now, as I just mentioned, our DNA polymer does have a directionality to it. So our 5' end is unique from our 3' end. So again, you can think of this as analogous to our N-terminus and C-terminus of a protein. So at our 5' end, we have our free phosphate with its negative charge. At our 3' end, we have a free alcohol where we can continue to attach more nucleotides. By convention, we always read our sequences from 5' to 3'. So the 5' is considered the start of our sequence, and the 3' is considered the end. Our DNA and RNA polymers look quite similar. So of course, they both have the same sort of phosphodiester linkages between them. Again, the only key difference is just the presence or absence of that extra hydroxy group at the 2' position, and then the presence of uracil instead of thymine in our DNA. This small difference between our DNA and RNA does lead to a significant difference in the behavior of our RNA versus DNA, right? So our extra alcohol here leads to additional interactions and reactions that we don't see with our DNA. So it does make our RNA more flexible. So we'll see some advantages of this later on. But it also increases the reactivity of our RNA towards hydrolysis. So this extra alcohol here can actually help to catalyze a hydrolysis reaction under basic conditions. So you can see if we have a base here, we can deprotonate our alcohol and then have a nucleophilic attack, which will break off our chain here and result in this cyclic phosphate derivative here. So this reduced stability of RNA, of course, illustrates the importance of using DNA in that genetic role, where it's more of a long-term carrier of genetic information, whereas our RNA is more just a temporary short-term role carrying that genetic information from our nucleus to our ribosomes. So the investigation into DNA structure did not stop once it was discovered that our nucleotides were linked together through phosphodiester linkages. Rather, research continued to try to determine a 3D structure for our DNA. In the 1950s, Rosalind Franklin and Maurice Wilkins looked at DNA structure using X-ray diffraction. Now, of course, we've seen X-ray diffraction and crystallography used to look at protein structure, and the same general principles apply to using it to study DNA structure. So in their X-ray diffraction experiments, they saw a series of bands from X-ray scattering from the DNA crystal. So you can notice all these dark spots in a very regular pattern around our center point here. So they determined this cross shape of the bands represented a helix, while they used the positioning of the darker bands on the outside to predict where the sugars versus the nitrogenous bases would be positioned. So based on this X-ray diffraction experiment, they again proposed the cross meant the helix pattern of our DNA, while the diamond structures meant that the phosphate sugar backbone is on the outside, while the nitrogenous bases point to the inside. They're also able to calculate key details about the helix, such as the width and the length of the helix based on that X-ray diffraction pattern. Now, Watson and Crick took this a step further. They used the X-ray diffraction data along with the other previous knowledge, and they proposed that the missing layer within the X-ray diffraction data suggested that there was an alternating pattern of major and minor grooves in the DNA. They further proposed that there was hydrogen bonding between the nitrogenous bases of our DNA. So they suggested that adenine was pairing up with thymine and guanine was pairing up with cytosine. This was based on earlier research from Erwin Chargaff, where he found that the amount of adenine in DNA was always equal to the amount of thymine, and the amount of guanine was always equal to the amount of cytosine. Right? So this precise ratio was discovered just based on chemical analysis of DNA, and that ratio was then used to inform the proposal of the 3D structure. So based on this pairing, along with the helical pattern, Watson and Crick then proposed a double helix model for our DNA. So the double helix fit both the older data showing that the pairing between nucleotides as well as the new X-ray diffraction data. This double helix model led to the Nobel Prize in 1962 for Watson, Crick, and Wilkins. Unfortunately, Rosalind Franklin did not receive the prize as she passed away before the prize was awarded, and they didn't award it posthumously. So the Watson-Crick 3D model of DNA, then based off of that X-ray diffraction data from Wilkins and Franklin, led to this 3D structure shown here. So we have our two strands wound together around each other, creating this a very typical double helical structure, which I'm sure you've seen before. We have our bases on the inside marked in yellow, and you can notice these blue dashes as they are hydrogen bonding together. We also have this pattern of major and minor grooves here between our helices here. So you can see that clearly in the structure over here on the right. So you can notice you have this major group. We have this large gap between our sugar phosphate backbone, where our DNA nucleotides are a little bit more exposed there. And you also have this narrow gap. That's our minor groove, and these will alternate. So you can see it's minor, major as we move along our DNA structure. This double helix model of DNA that Watson and Crick proposed is also called B-DNA. Again, of course, the center of our helix is formed by our nucleotide bases, which are pairing up through hydrogen bonds. And since we know the structure of our bases, we can actually see the individual hydrogen bonds that are forming here between our bases. So remember, we had a specific pattern of hydrogen bond donors and acceptors on each of our bases. So we have one key hydrogen bond donor on each of our thymine and adenine. And we also have a hydrogen bond acceptor that pairs up with our hydrogen bond donor in order to form our hydrogen bonds here between our adenine and thymine. When we look at guanine and cytosine, we see three key hydrogen bond acceptors. We have two on cytosine and one on guanine, and these are pairing up with three key hydrogen bond donors. Because of this specific pattern of hydrogen bond donors and acceptors, we can only pair adenine with thymine and guanine with cytosine, right? So you'll notice our adenine has this pattern of donor and acceptor. That would only match up with another base with the exact opposite pattern of an acceptor and then a donor. For our guanine and cytosine, our pattern here is acceptor, donor, and donor, which would only match up with the opposite pattern of a donor, acceptor, and an acceptor. So it's actually because of our hydrogen bonding that we have this specific pairing between our nucleotides. You can also notice that our guanine and cytosine link together with three hydrogen bonds, so it actually forms a tighter interaction compared to our adenine and thymine. So it's actually easier to break the interaction between adenine and thymine versus between guanine and cytosine. You also might remember from the beginning of our course that our hydrogen bonds are highly directional. So they are stronger in a linear arrangement. And so this actually helps to increase the strength of our base pairing here. So notice how we have a straight line between each of our hydrogen bond acceptors through the hydrogen and the electronegative nitrogen of our hydrogen bond donor. So all of these are aligned in straight lines to create these strong hydrogen bonds here, and any shifting of our bases would disrupt that strong interaction here. So this actually plays a key role in helping our DNA to link into that specific structure, because we have to get our bases to pair up in the right formation to get those strong linear hydrogen bonds between each of our bases here. So our two chains will differ in sequence, again, reading from 5' to 3', but they will be complementary, right? Meaning that we always have to pair up our amino acids based on the hydrogen bonds that are forming there, right? So we'll always have a guanine in exactly the opposite place as our cytosine on the other strands. We also see that our chains run anti-parallel to each other, right? So one chain is running from the 5' to 3' end, top to bottom, whereas the other chain is running from 5' to 3', bottom to top. So our chains are actually running in opposite directions, anti-parallel to each other, and then pairing up against each other according to that. So if we read our sequence, right, on one side we'd have a C at the 5' position. On the other side, we'd have T at the 5' position. So their sequences wouldn't initially look like they're complementary if we're reading them both 5' to 3', but once they're placed anti-parallel, that's then when we get to see how our bases pair up. In looking at this complementary nature of the DNA, Watson and Crick further noted in their 1953 paper that it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So since each of our bases are complementary, and each of our strands then are perfectly complementary, we can actually predict and create our second strand from our first strand. So this is the process of DNA replication. In order to replicate our genetic code, we simply have to separate our DNA strands, and then each of our strands will provide the exact template that we need to create our new strand. So we just have to copy each of our original strands to form a new daughter strand, we call it, based off of the pairing between our nucleotides, right? So wherever we have an A, we'd end up with a T in our new sequence, and that will end up being exactly the same as the opposite strand from our original parent double helix. Now, of course, we will need enzymes to catalyze this, so there are our DNA polymerases that help to catalyze this reaction as we replicate our DNA. But the end result is that each of our parent strands gets copied to make a daughter strand that is identical to the other original parent strand. So they each end up making that complementary sequence, and together they end up perfectly replicating the DNA double helix. This complementarity is a key component of the central dogma of biochemistry. So the central dogma of biochemistry states that our DNA replicates itself and then can be used to create complementary RNA, which is then used to create all of our proteins. So we use that complementarity of our DNA to replicate our DNA so that we can pass it on through our generations. We also use that same complementarity to copy our DNA and make messenger RNA. So this is the process of transcription. We can then take that mRNA and use it to carry our genetic code, since it's a perfect copy of the complementary sequence of our DNA. We carry it into our ribosome, where we translate it now into our polypeptide or protein chain. So that then results in our unfolded protein. We do have other RNAs involved here in our translation. So our tRNAs are involved in that actual translation process, whereas our mRNAs are the messengers carrying that genetic sequence to our ribosome. And of course, as we've seen in our previous discussions, that unfolded protein chain then has to be folded up into our specific protein structure to create then our active protein that can carry out its role in our cells. If we take a little bit of a closer look at our transcription, we can also see the same complementarity involved here. So instead of a DNA polymerase that we use to copy our DNA, we have an RNA polymerase that will create our RNA from our DNA template. So we have our polymerase, of course, separating our strands. So you can see our strands get pulled apart to get our single-stranded DNA. And then we're able to bring in our RNA monomers. So we have our RNA nucleotides coming in the form of nucleotide triphosphates. these react and are added on to our growing RNA polymer as we create that mRNA messenger. Now, while the molecular structure of our DNA and RNA makes this all possible, it's important to note that proteins are also critical for binding to our DNA and RNA and controlling our replication, transcription, and translation processes. So while the chemical structure of our DNA and RNA is critical, we do also require proteins as well. In all of these different interactions between our protein and our DNA, the shape of our DNA is, of course, going to be critically important for determining what that interaction looks like. Now, so far, we've only been looking at this typical Watson-Crick double helix B form of DNA. But in fact, our DNA can fold into other structures as well, right? So just like with our proteins, how we can fold up into all kinds of different unique shapes, our DNA will also be able to fold into different unique shapes as well. Now, we do have much less variety of shapes with our DNA compared to proteins, but there are still some important differences in our shape. So in our next lecture, we'll look at these other forms of DNA structure, and we'll see how they influence the interactions between our DNA and proteins, which are so important for our transcription, translation, and replication processes.